I'm going to talk about our iCell cardiomyocytes, which are iPS-derived cardiomyocytes, and then some of their uses in the cardiotox assessment. As an overview, I'm going to give an introduction to our company, as we are a fairly new company, and our iPS cells and the derived iCell cardiomyocytes, as that's our flagship product that has been launched. And then we're going to look at the cytotoxicity assessments using the cardiomyocytes and some of the Promega assays. All right, so our founders, we sprang out of the UW, University of Wisconsin at Madison, and sprang with a strong expertise in stem cells. It creates our foundation. And as our first product, cardiomyocytes, we've got practicing cardiologists from the UW lab as well. Tim Camp worked with Jamie Thompson 7 to 10 years ago, developing cardiomyocytes from the stem cells. Craig January has been involved in the cardiac arrhythmia for roughly 2 to 3 decades. I can say that because he's not here, so it's been several years. He's one of the pioneers of the field. Secondly, the way our this high level of talent up top attracts a strong lower level talent as well. And the way that we've got our company arranged is it's research, cell biology, R&D, process sciences, and then a production. And we've got a nice influx of scientists from several of the leading institutions in the country, both University of Wisconsin, Medical College, National Jewish Center, several private institutions as well, including Promega and EMD and some other good companies. So we've got a real strong industrialization-oriented way of making our cardiomyocytes. Second thing is about the iPS cells versus embryonic stem cells. We are purely induced pluripotent stem cells. And what this is, we take an adult sample and then we reprogram that by introducing the various genetic factors. That makes our master cell bank of stem cells. Those can then be differentiated into any of the different cell types. As long as the science is there to differentiate them, we can do it. What this leads then is that this graph is to represent that we've got any of the human cell types we can make on the y-axis versus really any member of the population. So we can make stem cells from anybody, anywhere. And the potential uses here, first low-hanging fruit is tissue toxicity, cardiomyocytes or hepatocytes, neuronal cells. You can also generate population-specific panels, looking at ethnicity, predisposed populations, clinical settings, so LQT patients that have genetic diseases that can be recapitulated in the dish and then studied. Like I said, we can do disease models. We can also engineer in disease models. And last but not least is the drug discovery. So it's really a basis for the next wave, the next future of technology discovery and toxicity testing. In CDI, quickly realized that we're not just going to be able to kind of sit at the bench and make a product here or there. We really wanted to get a foundation from which we can build. This is, I think, very similar to the Promega's luciferase system, where they've got a rock-solid system that can then branch off all of the products. We ended up making a process that we apply similarly. And that process is that we'll take the stem cells, we do a genetic engineering of them, putting in selection-specific markers controlled by gene-specific promoters. That happens out here. We then industrialize the process of making stem cells. We culture over 1 billion stem cells a day at CDI. We then differentiate those cells into the tissue-specific cell types using the various proprietary techniques, and we can get anywhere from a 10 to a 50% purity at that level. We can then apply our antibiotic selection that's under the control of a specific gene to generate pure cardiomyocytes, hepatocytes, or neuronal cells. At this point, our flagship product is cardiomyocyte cells, and that's what I'm going to be talking to you about today, and we're going to just end with a little teaser on the hepatocyte cells. In the cardiomyocytes, what this ends up is that we have the cells are grown in suspension in aggregate form, and before selection, we're sitting here with many aggregates, and you can see that we've got an RFP marker on the cardiomyocytes. Not all of the aggregates, so you can see these bunches in here, are not lighting up. So those do not have cardiomyocytes in them. We're sitting at about a 13% purity here. When we apply our antibiotic selection, that purity then jumps up to 98%, where right now we're below the resolution of the fax machine. We're at basically 100% purity at this point. You can see a 1-to-1 correlation. Each aggregate is composed of cardiomyocytes. What this leaves us with then is a population of cardiomyocytes. Here, these are cells that are grown in suspension, They're grown and they appear as just the beating aggregates. Each aggregate has about 500 to 2,000 cells beating in it. They are labeled with RFP. They're generated from iPS cells. They're 99% pure. We've got a colorless version that will be coming out at the end of Q1 here. Next, I'd like to go into just a little bit of the characterization of these cells, and then we'll move into some of the promega kits, some of the cytotoxicity that we looked at. These cells express all the appropriate markers that we've looked at so far. Stem cell markers go down, so what we're looking at here, let me back up a second, are qPCR assays. Relative expression on the y-axis, time of differentiation on the x-axis. Day 0 is stem cells, day 11 is during our differentiation process, and day 32 is our release age. So we see stem cell markers go down, appropriate transcription factors for cardiac cells increase, then later on we see an increase in ventricular cardiomyocytes. So these cells are maturing as they differentiate. We also see the upregulation of various cytoskeletal or myofibrillar proteins, actins, myosins, and troponins. We also look at the heart cells beat. That's their primary function. And to beat, you need an ion channel to convey that electrical impulse. And we see appropriate ion channels here as well. We've got sodium channels are upregulated, calcium channels, which are responsible for the plateau phase, are upregulated, as well as potassium channel family members, KCNH2, which is hERG, KCNQ1, which is the slowly delayed repolarizing factor channel, and its beta subunit, KCNE1. All of these are upregulated in an appropriate fashion. We also look at, very briefly here, at protein characterization. Here we just see a phased picture of cells 24 hours post-plating. They haven't yet begun to form their syncytia yet. And they all have the NKX factor. So they're all identified as cardiomyocytes. On the lower panels, we see cells that have been in culture for several days. And we can see the formation of the contractile apparatus. Here we stain with sarcomeric alpha-actinin. And over here, troponin. Troponin is more widely expressed, but we can also see, if you use your imagination, you can see some of the striated banded pattern there. The next 3 slides are going to show various currents. Here we just see sodium channel currents, typical waveform. These channels are activated by voltage, and you see the appropriate voltage, current voltage response. Calcium channels are expressed at much lower levels, and so here we see under control conditions, inward current. We then block all of the calcium channels pharmacologically, then just do a subtraction to get a pure measurement of the calcium channels. And again, we see an appropriate current voltage relationship. The last slide we'll show on channels is the hERG channel. And some of you may be aware this is the channel that is blocked by drugs unintentionally and can cause ventricular arrhythmias and lead to death. It's been known for a long time, as we'll talk about in a minute, but it really came to light when people taking non-sedating antihistamines were having adverse events. At any rate, again, it's a small current in its native cells, and so we do a pharmacological subtraction, and we end up with a pure hERG current here. We've got an outward bell-shaped current during the step, and then a tail current upon repolarization right here. Again, exactly what you would expect from a native primary cardiomyocyte. What this results in, a family of action potentials. These were recorded with perforated patch. And what you can see here is that they recapitulate the types of action potentials that you would see in the adult heart. In the adult heart, you'll have nodal action potentials, atrial action potentials, and ventricular-like action potentials. We see in our cell populations atrial-like, nodal-like, and ventricular-like. So we've got a nice recapitulation of the electrophysiology of the heart in these cells. This slide just shows that they're pharmacologically active. Here we just see a train of action potentials that's been recorded. Under normal conditions, you have your typical cardiac action potential. You add a little bit of E4031 at physiologically relevant levels. This blocks the hERG channel. It'll prolong the action potential and generate these arrhythmic triggers called EADs. You can hang the cells up at a depolarized potential. This is very bad. If you wash the drug out then, your channels return to normal. If you block calcium channels now, which are involved in beating, you completely cause the spontaneous beating to stop. If you wash out, you return to normal. So you've got a nice system here that recapitulate cardiac physiology. Now I'd like to move into some of the applications, focusing on a little bit, real briefly, on the ion channel electrophysiology, and then going into the cytotoxicity assays. Really what I wanted to point out is that the drug-induced cardiotoxicity is not a new problem. This upper graph here represents an EKG tracing of a patient at the University of Wisconsin-Madison who was placed on estemazole, which is the trade name of Hismanol. She has a normal QT interval, but then enters into some arrhythmogenic activity, and that becomes what's called a torsade du pont, or a twisting of the point. If this happens to you and you're not in the hospital, you're most likely going to die. This is around, as you can see, 1996, but this type of ventricular arrhythmias has been well known. Here's a similar quinidine effect that was recorded back in 1976. So the cardiac arrhythmias have been well known. The drug-induced cardiac arrhythmias have been well known. Biochemical-induced events, to me at least, are a little newer. And these will include things like cell permeability, energetics, oxidative stress, mitochondrial dysfunction, necrosis, apoptosis, you name it. And here these are typically things that occur in the body after an amount of time. These will happen as soon as your compound or your drug hits its Cmax. These tend to happen after accumulation. And so my point here is that these two toxicities are not necessarily causal or linked to each other. So a lot of people want to see dual assays that will attack both of these. If a drug causes an arrhythmia, that doesn't mean it's going to cause any type of a cell death or a biochemical reaction, and vice versa. And that was my tagline for this slide. All right, so for the iCell cardiomyocytes, I showed you some slides of beating aggregates. We actually ship them as cryopreserved suspensions of single cells. These are shipped as vials that are in unit sizes to plate 1 or 5 96 well plates. These are a 1x or a 5x unit. We warranty that there will be at least 1.5 million platable cells. So you can thaw your vial and plate a 96 well plate at 15,000 cells per well, which will give you a fairly confluent monolayer, which will beat as a syncytium and allow you to attack the electrophysiologically relevant and biochemically relevant processes with your assays. The way we do that is we take into account plating efficiencies and cell loss upon thawing. We basically overfill the vial to guarantee that you have enough cells. The general flow for the cell-based assays is that one would thaw the cardiomyocytes, plate them into your plate with supplied plating media. After day 2, you change the media, and then depending on the needs of your assay, you could begin the drug incubation anywhere from days 2 to 6. It's really up to you. And then perform the assay between days 3 and 7. The image up here are just images showing an entire well of a 96 well plate, plated at, here I believe it was 5, 10, and 15,000 cells, showing that as you increase your density of cells, as one would expect, you get a nice increase in density of the cells that have been plated. And this graph illustrates the purity of the cells as they're maintained in culture, and this is really a crucial point here. We supply maintenance media that will keep your cells pure. There's that slight, small percent of dividing cells in the culture, and if left unchecked, those can, over time, take over the culture under typical culture conditions and then skew your assay results. Here, if you use the iCell maintenance media that we provide, your cardiomyocyte cultures will remain pure. This shows we have a high level of purity maintained, virtually no change, over 2 weeks in culture. This is plated on the cell. Whereas if you'd use just a typical maintenance media, or typical cell culture media, you can see a decrease in purity of about 20%. Now you may not be sure if your compound is hitting these cells or your cardiomyocytes. So really the use of our highly pure population of cells and our maintenance media that guarantees a high purity is maintained, you can be sure that the results you obtain are due to an interaction with cardiomyocytes. All right, next I want to like to look at some of the assays that were done both at Promega with Chad Zimbrick and at CDI with Maya Firstenau-Sharp. First point is that the cardiomyocytes are a receptive test system. Here we see just cell titer glow looking at viability over a wide variety of classes of compounds. Compounds are listed here. We've got a wide class variety here of alkaloid protein kinase inhibitors, anthracyclines, quinozolones, and ionophore, tyrosine protein kinase inhibitor, and then an agonist, which is isoproternol. We've got the targets, or the mechanism of actions, and the EC50s. And so you can see we can get good data from a wide range of actions, coming down here from the sub-nanomolar range up to the hundreds of micromolar range. And these cells were pretty much plated as iCell cardiomyocytes in the 96-well format, did a serial dilution of test compound, we've got a 1% DMSO at all concentrations, then we've assayed with the Promega cell titer glow after a 24-hour incubation. You'll see this methodology just giving you the specifics that's repeated on each slide. So we've got cells are sensitive to a wide range of compounds over a wide range of IC50s. Here we get into the multiplexing, looking at the multi-tox glow kit, where in this case, we've applied staurosporin, and we see the loss of viability that's correlated with an increase in cell death. The cells aren't just being washed away, they're actually dying. So we see a good concordance there. We also see here with imatinib, which is Gleevec, again, nice concordance between the loss of viability and the increase in death. Second point is that the cardiomyocytes are responsive across a whole host of different assays. We've got apoptosis, caspase glow, 3-7. We've got the GSH glow, oxidative stress, proteasome glow, here using the positive control, and then the HDAC glow, soon to be released kit, using the positive control. Nice dose-dependent relations in all of these. So we've seen some phenomenology, and we've seen class effects, and now we can actually dive into the mechanistic effects. So here we have staurosporin. Again, looking at cell titer glow, you see a loss of viability. With the caspase glow, we see an increase in apoptosis or caspase activity, so an increase in apoptosis. So here, cells are dying through apoptosis. We apply Gleevec, you see a loss of viability, but there's no activation of the caspases, so the cells are not dying through caspase activity. We've gone into a mechanistic insight here. Staurosporin induces a decrease in viability linked to apoptosis. The other one is independent of apoptosis. Those have all been in 96-well plates. We're moving to miniaturizing to allow higher throughput assays, and here's one of our first forays into that, and we've used the 384-well plates, and here we've just done the cell titer glow with a staurosporin and a variety of concentrations. And here we've got cells that have been washed, meaning the pipette has gone through and exchanged the media, or you've just flicked it. Either way, you get very similar IC50s or EC50s that match what you saw in the 96-well assay plate. So this system is very amenable to miniaturizing and moving into really higher throughput primary or secondary screens. In summary, for the cardiomyocytes, we've manufactured these through a controlled industrialized process. We've got that foundation that includes the genetic engineering, tightly regulated process and manufacturing system. We manufacture billions of cardiomyocytes, and we're in the process of now moving on to the different cell types. Our cardiomyocytes show the appropriate cardiac gene expression, function, and responsiveness, as one would expect. They perform as expected when used in typical viability kits to assess various cellular processes, including viability, apoptosis, oxidative stress, histone deacetylation, and the proteasome function. Really, anything you want to look at, we'll be able to look at with these cells. And both of them combine to provide a really easy, user-friendly assay system. What I'd like to leave you with here is just a little teaser, so it's a little off track here. And this is in our hepatocytes. These have passed by our feasibility gate, and we are in the process of looking at collaborations with people to work together so we know what you need so we can engineer these cells and develop something that's going to be really useful. We don't want to do the if we make it, they'll come. We want to make something that's going to be very useful for you. Our feasibility for our hepatocytes we're looking at a molecular identity of several markers, protein function here, and then our experimental controls are HepG2 cells and primary human hepatocytes. Right now, we're just out of the gate without any genetic engineering. We're at a purity of about 15%, and we can see the transcription factor and albumin production being expressed in a nice well of hepatocyte-like looking cells. Tightly adhered, nice cobblestone morphology in there. If we look at our gene expression, we can see that the methods that we're using in general fall between the primary human hepatocytes here and the HepG2 cells here. So we've got just out the gate a nice bar where we're already surpassing a HepG2, and we're approaching that of the primary human hepatocytes. We can see that in protein secretion here, the HepG2, and down here with the CYP3A4 induction that was measured with the P450 glow. We can see the error bars here are very large because in our recent runs we've been getting induction that's closer to the human hepatocytes just by manipulating the system using that deep knowledge that we have in stem cells and the stem cell differentiation. These will be available for late summer for early access. Looking for collaborative partners right now. There are several CDI folks in the audience that would love to talk to you if you have any ideas. If anybody has any questions or comments on either of these items that I've talked about. Thanks for your attention. I'll take any questions.
So you've demonstrated that in a vial you can basically place a platelet, 98 well or whatever.
Yep.
What is your reproducibility between vials?
Between vials, I can say it's very reproducible. I don't have the data here to show you, but we've run these across vials in a lot. Part of our QC is as these are vials, we look at beginning, middle, and end, and then we've also done many experiments across lots as well. And that's the reproducibility factor there. I mean, we don't see any more inter-lot variability than there would be intra. And this intra is well below a factor of 3, so we're fairly satisfied with that.
For neuronal cells.
We've got a pan-neuronal population right now that, again, is also out of our feasibility gate. So we've made GABAergic, serotonergic, dopaminergic. It's really now a matter of defining what the customer demands are before we go to the next step.